A spectrophotometer is an instrument used to measure the intensity of light absorbed or transmitted by a sample as a function of wavelength. It is widely used in chemistry, biology, and material science to analyze the concentration or properties of substances. The principle behind the spectrophotometer is the Beer-Lambert law. This law states that the absorbance of light by a sample is directly proportional to the concentration of the absorbing substance and the path length of the light through the sample. The formula is absorbance equals molar absorptivity times concentration times path length. Where absorbance represents the amount of light absorbed, molar absorptivity is a constant for a substance at a specific wavelength, concentration is the amount of the sample, and path length is usually 1 centimeter in standard cuvettes. The instrument measures how much light is absorbed or transmitted at specific wavelengths. This allows for quantitative analysis, such as determining the concentration of a solute, or qualitative analysis, like identifying substances by their absorption spectra. The main parts of a spectrophotometer include the light source, monochromator, sample holder or cuvette, detector, collimating optics, and the readout or display system. The light source provides a continuous spectrum of light. For ultraviolet visible spectrophotometers, a tungsten filament lamp is used for visible light, and a deuterium or xenon lamp is used for ultraviolet light. For infrared spectrophotometers, a heated ceramic or nichrome wire is used. The monochromator disperses light into specific wavelengths using a prism or diffraction grating, allowing selection of a narrow wavelength band for analysis. The slit in the monochromator can adjust the resolution. The sample holder, or cuvette, holds the sample in the light path. It is typically made of quartz for ultraviolet visible measurements or glass and plastic for visible light. The standard path length is 1 centimeter. The detector measures the intensity of light transmitted through the sample. Common types include photomultiplier tubes, photodiodes, or charge-coupled devices for high sensitivity. Collimating optics, such as lenses or mirrors, focus and direct the light beam through the system, ensuring a parallel beam for accurate measurements. The readout or display system processes signals from the detector and displays results as absorbance, transmittance, or concentration. Modern systems often include digital interfaces and software for data analysis. The working process is as follows. Light from the source passes through the monochromator, which selects a specific wavelength. The monochromatic light travels through the sample in the cuvette. The detector measures the intensity of light exiting the sample. The system compares the transmitted light to the incident light to calculate absorbance or transmittance. Results are displayed or recorded for analysis. Applications include quantifying DNA or protein concentrations, measuring solution concentrations in clinical diagnostics, and characterizing materials such as pigments, dyes, or nanoparticles.